Thank you so much, my friend, for your kind introductory words. And yes, indeed, for 30 or 40 years, my whole family with the grandchildren comes to Greece to spend 2 weeks of vacation. Obviously, I have many friends in Greece and I come to meetings over the years. But Corona hit us hard. And for the last 2 years, we didn't come to Greece, but we are planning for next year. Hopefully, this will be possible. And we always enjoy the hospitality of the Greek people and obviously the hospitality of all the friends that we have in Greece. Now, I was asked to address the issue of biosimilars, and biosimilars are not a new concept. They're around for almost 10 years in the field of rheumatology, but it is something that leads to talking, to thinking, to asking questions. Here is my disclosures. And so not infrequently, I'm asked how I treat rheumatoid arthritis. Now, trust me, I don't treat it differently than any of you. And while in an Italian proverb, se non è vero è ben trovato, the assumption that I know as much as all of you together is really not my assumption, and I don't think so, because all of us know a lot about rheumatology. But when I think of what governs my approach, then obviously I don't need to tell you that it's the EULAR recommendations for the management of RA with the latest updates, and the latest updates was in 2019. But there is another point that governs my approach in general, in medicine in general, and that is the recommendation by WHO on the rational use of medicines. And what do they say? They say medicine's use is rational, and they define rationality as appropriate, proper, correct, when patients receive the appropriate medicine in doses that meet their individual requirements for an adequate period of time, and then they say, and at the lowest cost, both to them and the community. So it's always these three aspects, and these essentially govern my approach. Now today, we have sort of worldwide, maybe yes, biosimilars I didn't include here, 17 DMARDs available to treat RA, 3 conventional ones, and you can add hydroxychloroquine, glucocorticoids for bridging, and I didn't even include the glucocorticoids among the 17. We have 5 anti-TNFs. We have 2 anti-IL-6 receptor, 1 co-stimulation blocker, 1 anti-BCR. I don't need to tell you that we have 5 JAK inhibitors, 1 in Japan, pefisitinib, and maybe other places in Asia. And since several years, we have biosimilars for adalimumab, etanercept, infliximab, and rituximab, and more will come. With this array of possibilities, and this is the background of trying to really get, and we have treatment strategies, obviously, to really get the EULAR recommendations done, one needs order, and one needs some reshuffling. It's not like a play of cards where you say, well, for this guy, it's the king, which is one of the drugs, and there is the queen, and there is the jack, or whatever. And this is what the EULAR RA management recommendations try to do, and I will use them in some sense as an anchor, but I will focus on efficacy, and if we have safety issues to discuss, then we can obviously discuss them. So this is the latest version, and what EULAR suggests is to start with methotrexate in combination with short-term glucocorticoids. There's lots of reasons for this. I'm not going to allude to them. Lots of reasons for this. No better drug than this combination. No biologic, nothing better than this combination. But if that fails, we still need to address remission in every phase, and remission not by DAS28. That's useless. When you use IL-6 receptor blockers, even TNF inhibitors, not so easy. When you use JAK inhibitors, then DAS28 is useless. So we have the EULAR Boolean or index-based definitions with the SI and the CDI. We can discuss that too. I didn't include slides on that further. But when that fails, then we say add a biologic or a JAK inhibitor. And the combination is the true wonder drug. Now, back in 2013, with the 2013 update of the EULAR recommendations, the first recommendations were published in 2010, we already included, and we probably were the first organization in the world in rheumatology. When we mentioned TNF inhibitors, we also said biosimilars as approved according to a thorough approval process such as by EMA and or FDA. This is almost 10 years ago that we said that. We said they are equivalent because we had all the data that we needed available. And I know that there were discussions. We had also discussions in the task force. And there were many people who said, no, but show us the evidence of no. The systematic literature review showed the evidence of yes. So when we approach biosimilars, then the definition is obviously well known to all of us, a biotherapeutic product which is similar in terms of quality, safety, and efficacy to an already licensed reference biotherapeutic product with similarity defined as the absence of a relevant difference in the parameter of interest. Now let me stop here for a second. We need originator drugs. Originator drugs are bringing our field forward. It's the new science in the field, the new insights in the field. Remember how much insights we gained also with respect to pathogenesis when we learned that IL-12 and 23 inhibitors don't work for RA, but they work for psoriatic arthritis. We learn all the time. So originators are extremely important. But after 20 or 25 years, there are new originators. And this is when the world says, well, the patent is over. And then they should reduce the price. And this is when biosimilars come into place. So when you develop a drug, then you don't need much analytical study. You need to show that your drug binds something, and you need to show that it has efficacy in certain functional assays, like if we focus on TNF inhibitors, it binds TNF. It has efficacy when it binds TNF to prevent TNF actions on cells and others. And then for new drugs, for originator drugs, lots of clinical trials are necessary. Phase 1, phase 2, often 2 phase 2, phase 3. Usually the regulators require 3 phase 3 studies. Then there is phase 3, 4, phase 3B. Many trials are necessary. But as you can see from the title here, the regulators, not I, and probably also not you, realized already 20 years ago, 2004, 17 years ago, that there are changes in the manufacturing process, and the regulators said this has nothing to do with biosimilars. For the originators, the regulators said a determination of comparability can be based on a combination of analytical testing, biological cases, assays, and in some cases non-clinical and clinical data. That there was no biosimilar antibody around a receptor construct. This, the regulators, ICH, International Conference on Harmonization, that's what they stipulated for the biologic agents, because the regulators knew that there was batch-to-batch variations. And we were never told, not I, not you, none of us was told. So no batch of any biologic is identical to the other batch. And there are manufacturing changes, and look here, if you look at infliximab, then you can see that there were 50 manufacturing changes. If you look, whatever you look at, this is not only anti-TNFs, but whatever you look at, trastuzumab had 20 manufacturing changes, rituximab had 23 manufacturing changes, etanercept, adalimumab had manufacturing changes. And what does that mean? It means that certain changes also in the molecule occur. So what you can see here, this is from a paper from the Sandoz people in Austria. They have in the western part of Austria, they have their plant. And what they show, they collected hundreds of batches over the years. and analyzed them. And what they showed was, look at this chromatogram and look at this chromatogram. So often there are very little changes, but sometimes there are major changes. Look at the glycosylation. This is some batches, and this is all of a sudden a different set of glycosylation situations. And I could show you more of these. So when there are manufacturing changes, then, as we discussed before by the ICH, they ask for the regulators ask for analytical studies. And they ask again for functional assays. They usually, if these are okay, don't ask for clinical trials. As I mentioned, the producers of biosimilars have over the last 10, 15 years bought batches, hundreds of batches, and they check functional assays or other assays like here, and this is the batch-to-batch variation. Then there is a manufacturing change, so there is some other batch-to-batch variations, and what they try to make their product is right in the middle, right in the center of these batches, so that the variability can be reduced. But once again, batch-to-batch variations occur with the originators and not insignificantly. So when the biosimilars came out, and the first one was infliximab, the rheumatologists embraced it. I showed you what Euler did. ACR was more hesitant and had a sort of a very hesitant statement originally. But our colleagues from gastroenterology, they were heavily against it because it had not been studied in Crohn's disease. And what EMA and other agencies say, well, if you show us that this is the same molecule with the same properties, the same functional assays, then one study in one indication is okay and you get the rest of the indications. Why should an anti-TNF be different if it's the same molecule, more or less, in the batch-to-batch variation situation? But the ECHO people, the Crohn's group people, made a position statement heavily against having a biosimilar that wasn't studied in their disease. Now bear in mind, 6 months later, everyone had to use it because the hospitals wouldn't pay for the originator. So that rolled over immediately, more than in the rheumatology field. And they based their critique on the fact that the first or the second biosimilar had a slightly different antibody-mediated cytotoxicity than the originator. Now, you never have the identical things. No protein is the same if it is produced every day. Now, does antibody-mediated cytotoxicity matter for their disease? It doesn't matter really, because what you can see here, that this is an anti-TNF antibody, and you have a lot of ADCC by etanercept, which doesn't work in Crohn's. And you have no ADCC by certolizumab because it has no FC and it works in Crohn's. So ADCC is not important. They had the wrong argument. So no one has ever tested different batches of the originator molecules. I showed you variability, and I'll show you in a minute more variability. So Pekka Kurki, by the way, a rheumatologist working for the agency, wrote a letter back, and he said essentially no pharmacokinetic or safety issues are known to be specific to IBD, and the most responsive population, rheumatoid arthritis, was used to investigate immunogenicity, and therefore the clinical results enabled the extrapolation. And then the Northwich study came, as you know, and they used many diseases where TNF blockers are applied and showed that there was no overall difference between originator and biosimilar. I showed you this slide before, and I said that if manufacturing changes occur in originator molecules, then lots of analytical studies and functional assays are needed, but no clinical trial usually. And so how appropriate is it for the Crohn's people, or was it, now they all use it, for the Crohn's people to accept major manufacturing changes of the originator without demanding trials, but asking for the biosimilar companies to do specific trials, which would make a biosimilar by far more expensive. So let me show you another story. Trastuzumab, breast cancer. What you usually would see is this. In blue, you find the biosimilar. And in orange, you find the originator. So this is what you should be seeing. This is a break-off. But what they saw was in part a different situation. They saw that the originator was worse. And so all of a sudden, they could have been regarded as a bio-better, which would have made a totally different approach to approval. Because they had all the batches, they could show that there was a drift in the relative activity of antibody-dependent cytotoxicity and Fc receptor binding. So it was the originator that had the change. And because of the biosimilar study, this was detected. It wouldn't have been detected. And this is what I meant before. How appropriate is it when major manufacturing changes occur to not do a clinical trial to show that it's the same efficacy of the originator as the other one? I think that's an important question. You know, why do I say that's an important question? When we started to discuss biosimilars, the reps of the originator company said, it's not the same, it's not our process, it's not this, it's not that. But they never had told us before that they had changes in the molecule. As you can see here, that as I showed you with etanercept, I could show you with rituximab. So this is what made me a little bit surprised. All of a sudden, because biosimilars are coming up, manufacturing differences are discussed, but obviously only for the other companies, not for your own product. And this is simply not fair. And we should have been told better. The regulators knew. I showed you the ICH 2004, so they had discussed it already long before. I'll show you one of my studies with the Sandoz biosimilar rituximab. So here we studied GP 2030 and the European and the US rituximab, and you can see that the ESTA score is essentially the same all over the place. But look at this. When we looked at B-cell repopulation, what we saw was a major difference between the originators, between the rituximab from Europe and the rituximab from the US, and the biosimilar was right in between. It didn't matter clinically, but it could have mattered clinically. And this is the overall efficacy for all, and you see it's essentially identical. What about anti-drug antibodies? Everyone says, well, the biosimilar will have anti-drug antibodies because it's not produced by the original company. The difference was between Europe and the US, 21%, 13%, overall 15%. The same or is very similar to what was seen with the biosimilar. Now, biosimilars are interesting for the society and for every field, but also for the society because of cost reasons. And you see that some places in Europe take up rapidly. You know what? It's the richest ones. Denmark, Sweden, and Norway. Germany, slow, but finally it does it. Who are the lowest? Slovenia, Poland, you know, this is surprisingly. They should be more interested. This is infliximab biosimilars, that same story. Denmark was the first, and I believe, what is it here? I believe Italy is, interestingly enough, Norway, and then Italy is the next. So what are the cost reductions due to biosimilars for the hospitals, the societies, the field? Why am I interested in cost reductions? Not only because of our societal conscience. You know, I mean, our consciousness about society is important. You know, when you go to the grocery store and you see two very nice oranges and you don't see a difference between the oranges, you will probably take the cheaper one, especially if it comes from Greece. You wouldn't take the Italian one that is more expensive. So I mentioned Denmark, and in 2017, the approval was in 2013, 84% of the etanercept was biosimilar. And look at this. So here you have, first of all, this is the originator in dark. So that dropped more or less rapidly. And overall, what you see is an increase in the use of etanercept. But at the same time, you have almost half of the costs. And what about another, infliximab? Same story. Here, there is even a disappearance of infliximab originator from the market because they at least lowered the price. Merck didn't want to lower the price, and that's, I told them, they were with me. They told me, well, why don't you help us? Because we have given lots of deduction to the cardiology drugs. So I said, I don't have to defend cardiology money. I have to defend rheumatology money. So drop it by 20, 30%, and I will not change the therapy of my patients. Because the first ones were 30% lower. Now they are all 50% lower by law in my country. But what happened? That's the next story. Again, what you saw here, but even more. Look, infliximab use doubled over time. And this is when the second, this is the first, this is the second infliximab biosimilar. Look, and the costs were more than halved, despite the fact that it was double. So you essentially, per patient, it was 1/3 to 1/4 of costs. More people could get it. Now, if you take less affluent countries, poor countries, they should be interested in this because they can treat more people. I will come back to one story which makes me smile. But what about switching between originator and biosimilar? I know that some people say, well, I can think of using my anti-TNF as from the biosimilar, but my patients like my infliximab or their etanercept or their rituximab, and they don't want to switch. What about switching between originator? Bear in mind, as I implied, we all have already switched to not originators, different batches. And we don't know whether or not these batches had sort of, in some sense, slightly less efficacy or whatever. I showed you an example with trastuzumab. So here is a study where adalimumab was compared with this is adalimumab, reference adalimumab with biosimilar, and then after week 16, there was flip-flop switch in both arms. And as you will see here, no difference. First of all, no difference for the first 4 months, and then essentially you see here for a year, no difference in the 4 arms. They started slightly, at the end of the day, they get the regression to the mean because they are similar. What about more details? Is the PASI, this is psoriasis, by the way. PASI 50 and PASI 90, you can see that initially but this is after switch, and you see essentially the same. Another psoriasis study with etanercept. Here, again, etanercept versus, this is the reference, this is the Sandoz etanercept. Here are the arms, and they are continued, but then there is flip-flop. And you can see again for the PASI 90, initially the same, and then with the flip-flop, whenever curves, to me, I'm not a statistician. I don't understand much about statistics, but I like to ask questions about statistics. So whenever I see differences like this, I worry. I wonder what that means. However, when I see crisscross, then it's a play of chance. because here it's the other way around. And all of a sudden here it's the same. So you see crisscrosses or identical curves. This is an array study. Same story or similar story. Compare the Sandoz adalimumab with the reference adalimumab for 6 months and then switch the reference to the biosimilar. And you will see here, this is the change in DAS28 CRP. You will see here for the first 24 weeks, it's more or less superimposable. But then after the switch, there is also no difference, no loss of efficacy. By the way, adalimumab is, among all the proteins that I saw, the batch-to-batch variation has the lowest fluctuation. Now, there is one more problem, and that is, even if I would like to use the biosimilar because my hospital or my insurance wants me to use it. You know what happened to us once? After etanercept biosimilar was already approved, one of my colleagues in the clinic prescribed the originator. So he got a phone call from the payer, from the insurance, Social Security. And they said, Dr. XYZ, do you have a specific reason why you prescribe the originator and not the less expensive biosimilar? If you have a good reason, tell us and we will give it to the patient. He had no good reason, because just believes that's not a good reason. But very frequently the patients are reluctant, and that is the question. There's two possibilities. One, the patient doesn't want. Well, then you can tell the patient, you know what, if you want to keep the drug, you would have to pay for yourself. Then they very rapidly want it. There's no all of a sudden they don't have a problem to switch. But that's not the right way. It's just what happens, because if the payer doesn't pay, then the patient has to pay themselves for a drug. But the more important thing is to explain to the patients what we discuss here today. We call it framing, positive or negative framing. Mrs. Miller, there is a new drug out there. You know, it's not from the same company. I don't even know if it works the same way. Do you think we should give it to you? Then Mrs. Miller will say, no. But you can say it differently. Mrs. Miller, there is a new drug out there that's the same as your old drug. It just has a different name. And you know what? Over the years, there were many changes in this drug, and we accepted all these changes, and that's just a different batch, and it happens to make it also cheaper for you and the society. So we call it positive or negative framing. And you can see here from this study that the positive framing allows the switching much more frequently than the negative framing. And why is that? Because, and this is work from Italy, this is work from Fabrizio De Benedetti, who looked at MRI in placebo and in nocebo. That's a nocebo effect, because you don't change the treatment. And he found out that a positive expectation with a positive communication activates opioids, endogenous opioids, and endocannabinoids. You know, we are not addicted, all of us, but we know that opioids can make you high, can make you happy, especially the endogenous. Negative communication activates cyclooxygenase and cholecystokinin, and that makes you being afraid. If you want to read more, several years ago, 3 years ago, we have published a consensus-based recommendation for the use of biosimilars to treat rheumatological diseases, a large task force. And there is one more thing that's interesting. I don't know if you heard that. 2 days ago, the FDA gave a warning. Now, I use JAK inhibitors a lot, but the FDA gave a warning. I'm not sure that EMA will also do that. But the FDA said that these medicines, or the JAK inhibitors, should be reserved for patients who have had an inadequate response or intolerance to one or more TNF blockers. They explicitly said TNF blockers. I don't know why. I don't know why they didn't say Alzheimer's. So I'm just bringing this up. If you didn't hear about it, you will hear about it. I don't think EMA will stipulate the same thing, but I don't know. I don't know what the story is in detail because I haven't seen all the data. And we have registries where the JAK inhibitors behave differently than in the one study 1133 that elicited all this. But what I wanted to say here is that when they speak of TNF blockers, then also in the United States, there are biosimilars available. Let me close by looking again to the World Medicine Situation Report. It comes out every 10 years, so I expect a new one. I mentioned to you that they have 3 requirements. Meet appropriate medicines in doses that meet their requirements for an adequate period of time and at the lowest cost both to the community. And then they say irrational or inappropriate, improper, incorrect use is when one or more of these conditions is not met. And one of them is costs. And I think we all want to see innovative drugs. We all want to use new drugs for our patients. We're happy that the pharmaceutical industry produces new drugs for our patients. But the old drugs, when the originators don't drop, by the way, adalimumab originator made a tender in Norway because they were cheaper than the biosimilars. So, you know, this is what the government in Norway then decided, to continue using the adalimumab biosimilar, the adalimumab originator. But it's a question of price. So let me sum up, Mr. Chairman and colleagues. All BDMARDs have similar efficacy. I didn't show you that, but this is what we have seen. And JAK inhibitors are the least efficacious biologics and show efficacy across many IMiDs. BDMARDs should be combined with methotrexate to convey optimal results. The T2T strategy is target attainment at 6 months and leads to optimal outcomes. But then biosimilars approved in highly regulated areas, such as in Europe, are as efficacious and safe as originators, and switching from originator to biosimilar or between approved biosimilars is like using a different batch, such as due to manufacturing changes of the originator. Originators have not, or very rarely, performed comparative trials between batches of their BDMARDs. But overall, the prospects for RA patients become better with every year. We have good therapies, new therapies, good treatment strategies, and there is more to come. And with that, I'm happy to entertain your questions.